Good afternoon, students. Now we are going to study about the biostatistics. Coming to the introduction, biostatistics is a branch of biology which deals with facts and numbers. It is also called as biometrics. So it has a wide range of application in biology. So it is used in experimental work, research work like that. It shows a wide range of application in biology. So biostatistics is a science which deals with the development and application of most appropriate methods, like a collection of data. Then the collected data is presented in the form of a tabular column or a graph. Then it is the data is subjected to analysis and finally interpretation of the results. So after that, making decisions on the basis of results. So other definition for biostatistics is it is used to used in a referral to recorded data, denotes the characteristics and calculation for the set of data. Example, simple, mean, median, mode, like that. Then role of biostatistics. So to guide a design of an experiment or a survey prior to the data collection. So first, going for the data collection, we have to design the study. Then to analyze the data using proper statistical procedures and techniques, to present and interpret the results to researchers and other decision makers. These are the roles of statisticians. Then collection of data is from records or surveys or experiments. So if the data is collected through a survey, it is again divided into two types. One is comprehensive survey and another one is sample survey. So experiments mean scientific laboratory experiments like that. For example, estimation of oxygen in a given sample, or the samples collected from 3-4 areas like that. So these are the sources of data collection. So again, the data is divided into two types. The collected data is divided into two types. One is constant, another one is variables. So what types of variables? So there are two types of variables. One is quantitative, another one is qualitative. So quantitative variable is again divided into quantitative continuous variable and quantitative discrete variable. So under qualitative variables, it is also divided into two types: qualitative normal variables and qualitative ordinal variables. So next, methods of presentation of data. The data is presented in the form of numerical presentation, graphical presentation, or mathematical presentation. So what is numerical presentation? If the data is presented in the form of a tabular column, means it is called as a numerical presentation. So simple frequency distribution table is an example for numerical presentation. So next one is distribution of 50 patients at the surgical department of Alexandria Hospital in May 2008. According to the ABO blood groups, the blood group of a person is represented in the tabular column. So it is an example for numerical presentation. Next one is this is also an example for numerical presentation. So next one is complexity in frequency distribution table. So again, distribution table, here the distribution of 20 lung cancer patients at the chest development of Alexandria Hospital and 40 controls in May 2008. According to smoking. So here smoking, lung cancer cases are compared with cases are compared with control and also total. So like that, if the data is presented in a complex form, it is called as complex frequency distribution table. So this is also an example for complex frequency distribution table. Next one type of presentation is the graphical presentation. So the data are presented in the form of graphs. It is called as graphical presentation. So you all know that graph is presented by using graph paper. So it has an x-axis and a y-axis intersecting each other. So there are so many types of graphs: line graph, frequency polygon, frequency curve, histogram, bar graph, scatter diagram, or a pie chart, then cartogram. Like that, so many graphical presentations are here are available. So according to the data and also the investigator, the type of graph is selected. So this is an example for line graph. So data are plotted in a graph paper based on this on the tabular column. So the variables are connected by drawing a line. So it is called as line graph. So next one is frequency polygon. This is the tabular column for frequency polygon. So based on the tabular column, the frequencies are plotted and the frequencies are jointed by using the scale. So it is called as frequency polygon. Then frequency curve. So if the data are joined without using the scale, by if the data or the points are connected by using a free hand, means it is called as a frequency curve. Then histogram. So based on the data, we have plot the we can mark the values, and we can draw a bar diagram. So it is also called as histogram. So here a distribution of a group of cholera patients by an age is given as an example for histogram. This is a bar chart. So like that, two-dimensional view means it is the histogram in a two-dimensional view. It is called as bar chart. So it is another type of bar chart. This is an pie chart. So 360-degree a circle is drawn. So based on the observations, that circle is divided into various parts, and it is named by dividing with a percentage, means it is called as a pie chart. Then this is an example for a donut chart. Next, the mathematical presentation, summary statistics. So measures of location. So measures of central tendency, measures of non-central tendency, these are comes under measures of location, and measures of dispersion. So measures of central tendency means average, mean, median, mode. These all are comes under measures of central tendency. So mid-range is calculated by adding smallest observation plus the largest observation divided by 2. So we have to add a smallest lowest limit and upper limit divided by 2, means it we can get the mid-range. Then mode, the value which occurs with the greatest frequency, that is most common value. So number of times if the same value occurs, means it is called as mode. Next, median, the observation which lies in the center or middle is called as median. So mean means sum of all observations. So we have to sum the number of total observations, and we have to divide it by number of observations, means we can get the arithmetic mean value. So measures of dispersion. It is 5 types. First one, range, variance, standard deviation, semi-interquartile range, then coefficients of variation, finally standard error. So these are the measures of dispersion. Then this is an example for standard deviation. So standard deviation we will see in detail in the next class. Next, standard error. This is a formula for a standard error. So standard error, a measure of variability among means of samples and selected from certain population. It is called as a standard error. So thank you.